This is the fourth and final video I'm doing in a series of everything that's here on the board. In this video, I'm going to be talking about how ions cross the cell membrane. So ions, as we recall, cannot cross the lipid bilayer. They need a protein to facilitate their movement across, and those proteins are pores, channels, carriers, or pumps. Now I've drawn on the board here an example of a typical cell that you could find in your body. In this cell, we have potassium channel, sodium potassium pump, and you'll also notice that I drew here that sodium itself cannot pass the barrier. It cannot cross this barrier. It gets trapped on the outside. There are channels for sodium, but in the resting state in this cell, those channels are closed. Okay? So sodium gets trapped on the outside. In order to determine how easily an ion will cross a membrane, we actually have this thing called the driving force for the ion, which is the membrane potential minus the equilibrium potential for that ion. So the membrane potential is something that is a product of all the types of leaky channels that the cell had, whatever the cell expresses. So that's going to vary from cell to cell. It can be experimentally determined. A typical membrane potential is anywhere between -40 and -80 millivolts. Okay, that's pretty typical. The equilibrium potential is actually, that can actually be determined mathematically for each ion, and that's dependent on the concentrations of the ion in the extracellular and the intracellular space. And it's also determined by the electrical gradient, okay? So this is kind of something that takes those two and puts them into one place, because they're charged, they're attracted both to the opposite charge as well as whatever way their concentration gradient wants them to go. So there are two things that we have to factor in when we're considering where an ion will go. Now, why is this driving force valuable? It can actually tell us about the conduction of the ion, how it's going to work. So you may recall from physics way back when that when charges move along a wire, right, that you have this equation that you can use, which is I is equal to V divided by R, or V times G, which is essentially the current is equal to the voltage divided by resistance, or the voltage times conductance. Okay. Now, because ions are charged, they actually create a current when they go through channels. So we have this nifty little equation that we can use. The current created by the ion is equal to the conductance of the ion, times this driving force of the ion that we determined. Okay, and that will be useful later. We can figure out the conductance of the ion by the number of the channels, how probable it is that they're open, and how easily they conduct the ion through. Okay, so it's just a factor of how effective that channel is at moving an ion across, and that will tell us the current that can be created when channels are opened. We'll go into a lot more detail on some of these concepts in a different video. I just wanted to give you a brief overview of how ions cross the cell membrane. Last of all, I wanted to go over really quickly 4 types of pumps. We have F, V, P, and A, B, C transporters. Okay, so these are F-type pumps, V-type pumps, P-type pumps, and ABC transporters. F-type pumps and V-type pumps, those are both proton pumps. Okay, the F-type is found in mitochondria, and that's going to create ATP. The V-type is going to be found in vesicles, including in lysosomes, and that's going to create the lower, that's going to create the the lower pH that you'll find inside the lysosome. P-type pumps are the most abundant in the cell. They require phosphorylation to work. And these ones, ABC, and actually an example is a sodium potassium pump, they require the binding of ATP and subsequent hydrolysis to function. Okay, that's all for everything that I have here. I hope that this was helpful to you, and I hope you have a great day. Bye.